So over to introducing Sarah. It's my pleasure to introduce Sarah Marsh to this webinar this afternoon. Sarah is a product specialist at Atelerix. She has a background in immunology with a PhD in immunobiology. And with that, I will just pass on to Sarah. Sarah will be talking actually just before I do about BloodReady. The seminar title is No Bad Blood, Only BloodReady. And Sarah will be talking about a new product that has been launched this month, which is specifically for the preservation of whole blood at non-frozen temperatures. So over to you, Sarah.
Thanks, Steve, and thank you everyone for joining the webinar today. It's great to have so many people on the call. As Steve's just mentioned, this is an introduction to our new product for the preservation of whole blood. And please do provide any questions that you may have at the end of the webinar. So it's about 20 minutes, and then we'll have our FAQs. So we'll get started now. Hello, everyone, and welcome to Atelerix's latest webinar, hosted by myself, Sarah, and we'll later be joined by Steve Switlow, who is our Chief Scientific Officer for the FAQs. So this webinar is to provide you with an introduction to our latest product, BloodReady, which has been developed for the stabilization of whole blood samples. Rather than shipping fresh, you're able to extend the shelf life of your blood samples using this new product. So for anyone that hasn't heard of Atelerix or is new to us, we are a growing biotechnology company based in the northeast of England, in Newcastle. And our technology provides hypothermic storage solutions for a range of different biospecimens. So anything from blood, as I've just mentioned, to cells, to tissues, viruses, etc. And the major advantages of this are to mitigate the use of cryologistics. So obviously, cold chain shipment is a very expensive process. It's very damaging to the environment in terms of carbon emissions. And it's also not the most reliable method in that it requires high demand for dry ice. If there's any delays in shipments, samples can thaw, leading to a lot of stress and anxiety associated with shipping samples. In addition to this, our hypothermic storage solutions also provide an alternative to shipping samples fresh. So often sort of, for example, tissue biopsies and blood samples need to be shipped fresh. However, the quality will deteriorate fairly rapidly after collection. So it's really important that that transportation of the sample is a smooth process, which isn't always the case. So there is this demand for a solution, technology that can keep these samples fresh for longer without compromising the quality of the sample, but provides a lot more flexibility. So we're redefining how labs operate, trimming costs, and also being kinder to the environment. So how does our technology work? Just as a brief overview, following the principles of hyperthermia, so going from a higher temperature to a low temperature, at 32 to 37 degrees, cells are obviously under homeostatic conditions, metabolically active, and they have a fluid cell membrane. As the temperature decreases, though, below 32 degrees, the rate of enzymatic activity and subsequent metabolic processes in the cell slow down to such a rate that the cells essentially enter a state of hibernation. They're sort of in this paused state. And at this temperature, the cells are also very vulnerable to cell membrane damage, as you get this dysregulated transport of ions and water in and out of the cell. So ultimately, this will lead to cell death. And the way our technology works is by targeting this process. So our gels are able to stabilize the cell membrane integrity so that when the temperature does decrease to either room temperature or refrigerated temperatures, the cell membrane is intact. So you don't get this dysregulated transport in and out of the cell. The membrane is protected from damage, and the cells therefore remain viable for extended periods, from days to weeks. So because of this method of action of the acting on a lipid membrane, this means that our technology can be applied to any sample with a cell membrane. So it could be a small envelope virus, or it could be a larger cancer tissue sample, for example. So this has led to our existing portfolio of products. So currently we have a range of kits that are optimized for whatever your biosample type is. So for cells in suspension and viruses, we have CytoStore. We have TissueReady for tissues and organoids. For 3D models and adherent cells, we have WellReady. And then finally, for leukapheresis material, we have LeukoStore. We're always trying to expand our product range. And I know from my own experience that one area that would really benefit from our technology is stabilizing whole blood. So what are the challenges associated with storing whole blood? Well, obviously, whole blood samples are not very conducive to freezing, so that's cryopreservation isn't really an option. And the process of transporting blood is fairly inflexible, and this is because the sites of collection of blood are often distinct from the sites of processing and analysis. So transportation is required from one point from A to B. To make this even more inflexible, the actual quality of blood samples deteriorates pretty rapidly within the first 24 hours after collection. So as the blood isn't in vivo, it's not in its normal physiological state, a lot of changes occur to the blood, and this can lead to alterations in the expression of cell markers on cells. The relative proportions of different immune cells in the blood become skewed, leading to under or overrepresentation of different cell types. And ultimately, that leads to a declined analytical accuracy, which is no good for diagnostics, monitoring, any sort of assays looking at those cells in the blood. The worst possible outcome here is that the sample, if there's delays in transporting the sample and it's declined to such poor quality that the sample gets rejected and requires a recollection of a sample from a patient, that's pretty bad outcome. So other methods to overcome this inflexibility include various different fixative solutions. So commonly used are cytochecks and transfix, which do extend the processing time to provide some more flexibility with the workflow, but they're not the perfect solution in that fixatives themselves can actually can denature some epitopes on cell surface markers. Which can lead to inaccurate, for example, gating of cell types by flow cytometry. And fixation itself prevents downstream sort of functional analysis or proliferative assays as it kills the cells. So our solution is BloodReady. So as I described earlier, it's a hyperthermic preservation solution to extend the shelf life of blood. And using BloodReady, you can extend the shelf life from hours to up to 3 days. So it's really able to widen the time frame of opportunity to transport the sample, process the sample without compromising the quality of it or the sort of reliability of the data obtained. So how does BloodReady work? Well, firstly, you'll be provided with the BloodReady kit, and the kit will contain all of the components required to store the blood. So the kit will contain gel-based beads, a gelation buffer, and thirdly, a release buffer. So the process is it's simple, minimal hands-on time, really. So you would firstly add your whole blood sample to the gelée, which will take 30 seconds, 1 minute, and you'll then mix that solution to the gel-based beads. And mixing those two solutions will allow the gels to cross-link, and it will encapsulate the whole blood sample, and it will gel within 20 to 30 minutes, depending on the excuse me, the volume of blood used. Once the encapsulation process has happened, you're then able to either store a sample or ship it at either refrigerator temperature or room temperature. We would recommend sort of 2 to 8 degrees for the best possible results. At the receiving end, or when you're ready to use your blood sample, you would simply add the dissolution buffer, which is a simple salt-based solution. The gel will then dissolve within 15 minutes. And finally, there's a brief filtration process, just using a 100 micrometer filter to filter out any remaining dissolved beads. So altogether, if you were to carry out the process from encapsulation to release, it would take no more than 40 minutes. So it's very user-friendly and minimal hands-on time. So how well does BloodReady work? So as I mentioned, you're able to store your whole blood samples for up to 3 days. This initial data shows a comparison of the viability of CD45 leukocytes between non-stored, freshly collected blood and blood that has been stored in BloodReady for 3 days. So you can see that the viability is totally uncompromised. It's comparable with fresh blood. And the encapsulation and storage in BloodReady prevents any deterioration in viability for 3 days. In addition to looking at overall viability, we have looked at very thorough immunophenotyping of not only major leukocyte subsets such as B cells, T cells, and K cells, but also some of the more rare and intricate populations that are commonly of interest in immunophenotyping. So we've looked at, for example, regulatory T cells, gamma delta T cells, all the different subsets of B cells at different phases, so transitional, non-switched, switched. Some of the much smaller populations, such as dendritic cells and innate lymphoid cells. There's a lot of sort of pie charts and data here, but hopefully you can see that there's the proportions of these subsets do not change between freshly collected non-stored blood and blood that's been stored in BloodReady. This is really, really important for biopharma and clinical research organizations that are using blood samples to immunophenotype the blood to look at either disease or whatever the interest is. It's really important to know that the proportions are representative of that in vivo. The only difference we really see is in the granulocyte population. We do see a decline in the proportion of neutrophils. This isn't a huge problem. They're not typically a cell that are investigated in diagnostics as much as T cells, B cells. But saying that, they are still there, and you are able to preserve them, but there is this slight decline. Moving on, we have also looked at how well BloodReady is able to preserve the expression of a large number of epitopes on the surface of leukocytes that are commonly looked at in terms of their expression. So in this work, we've compared the ability of BloodReady to preserve epitopes with the ability of cytochecks, commonly used fixative, and compare the preservation of cell surface marker expression. So we have looked at a panel of 93 different markers, including those that are commonly investigated in research and in clinical research. So the expression has been divided into lymphocytes, monocytes, and granulocytes. And the expression of each marker here is expressed as a ratio of the expression in fresh blood. So 0 indicates equal expression to the marker expressed on leukocytes in fresh blood. So basically anything bright green is really good. Towards the red is increased expression, and towards black is a loss of expression. And what we found is that BloodReady is able to preserve over 90% of epitopes expressed on lymphocytes that have been screened, over 80% on monocytes, and over 70% of granulocytes. Comparing this with cytochecks, BloodReady is shown to preferentially preserve the expression of several cell surface markers better than cytochecks, including these markers here, CD62L, CD317, etc. So this is really exciting in that BloodReady is able to preserve these epitopes so well. And the other benefit here is that there's no fixation process with BloodReady, so any assays downstream are able to be performed even after the storage, which is an additional benefit over the cytochecks. So moving on, so we have looked at the ability to isolate PBMCs after storage in BloodReady for 3 days, to look at sort of the competence and function of the stored PBMCs derived from the blood. So looking at this here, we see that there's no real decline in viability of the PBMCs after being isolated from BloodReady. We've got the no gel control, where you can see the cells aren't as viable. They've not been protected in the same way, and the yield from cryopreserved blood is really poor, obviously. So that's around 12%. When we then look at the raw counts of different leukocyte populations, you can see that the raw cell counts are not significantly different from non-stored blood. In the no gel control, You can see that there is a decline in particularly B cells, T cells, and K cells. So what we're reiterating here is, that storage in BloodReady really provides this protective function of leukocytes for the 3 days without compromising viability. Then looking at how well the PBMCs recover overnight in culture and their response to stimulus. So looking at the PBMC recovery after they've been cultured overnight, we can see that PBMCs from BloodReady show 100% recovery. For the no-gel control, the PBMCs don't adapt as successfully to culturing conditions, showing poorer health and lack of competence compared to the BloodReady store PBMCs. We looked at the response to cytokine IL-2 stimulation, and you can see that PBMCs derived from BloodReady are very competent and respond to IL-2 stimulation after 2 to 3 days, whereas in the no-gel control, there's an absent response that's not present, unlike in BloodReady. So not only can you store your whole blood samples for 3 days, you can also then isolate the PBMCs and continue your workflow. So ultimately, BloodReady is able to extend shipping times by being able to store for 72 hours. It successfully preserves all major leukocyte populations and some of the smaller granular subpopulations, which is fantastic for flow cytometry. If you need a bit of more flexibility in processing your samples, it's able to conserve unstable surface epitopes better than common fixation methods, as we've shown with cytochecks. You're then able to isolate PBMCs with 90% viability using standard methods, maintain cell competence after return to culture. And BloodReady is available in multiple different kit sizes. So whether you're working with 0.5 ml of blood, 3 ml blood, or larger volumes of 10 ml, we can provide kits to suit your volume of blood. So just referring back to the challenges associated with transporting and storing blood, using BloodReady, you can really relax your workflow and relieve the stress and anxiety around working with whole blood, all while retaining the quality of the sample, maintaining accurate cell proportions, cell surface markers, providing you with analytical accuracy. The downstream applications are unlimited, and there's no worries about having to recollect a sample or have a sample rejected. So there's no bad blood. The only way is BloodReady. We do have a number of resources on our website. So the full white paper documenting all of the BloodReady data is available, and also protocols and price lists. So thank you, and I'd like to now welcome any questions that anyone has. Thank you.
Thank you so much, Sarah. That was a really good overview of BloodReady and a really enjoyable webinar there. We do have a few questions that have come through. If anyone has any further questions, please just pop them into the chat or into the Q&A even, and we will be more than happy to answer them. So, Sarah, just one that pops out here from Ruth. Does viability fall off a cliff after 3 days? Could you answer that one?
Thank you, Ruth. Seen your question come through. So we have looked at time points beyond 3 days. We looked at 5 days and 7 days to see how long the gels were preserving the leukocytes for at that 95% recovery rate. By the 5-day time point, the recovery dropped from 95 to around 70%. And then by the 7-day, 1 week period, it dropped further to around 40 to 50%. So we would recommend the optimum storage time to be 3 days, just to maximize on your cell yield and recovery.
Thank you, Sarah. And a question from Margot, is it provided sterile? I think I can answer this one. We are able to give full bioburden testing of our products upon release, so it can be provided sterile. Another question from Ruth. What is the autofluorescence of cells like after subjected to this treatment? Have you used spectral flow cytometry to analyze the samples to investigate this?
I can answer that, Steve, if you like. So the autofluorescence of cells, so we've only really compared it with the non-stored blood samples. Obviously, they haven't been exposed to the gel in the encapsulation treatment process. So I think assuming from that comparison, there's no minimal autofluorescence that's induced by the technology. I hope that answers the question okay, Ruth.
Thank you, Sarah. Question from Jennifer. Which species of blood is this compatible with?
Good question. As I explained in the webinar, the way the technology works is on anything with a lipid membrane. So although the data I presented was in human blood, it would be, it's applicable across the board of different species, blood samples. They've all got the same biological lipid membrane.
Perfect. Thank you, Sarah. So no cross-species differences should be anticipated. Bit of a tricky one from Ruth here. Oh, looks like a tricky question. Are the neutrophils still able to degranulate? Loss of newts is often because they have degranulated, so it would be interesting to see how you have actually measured what a granulocyte is versus a neutrophil versus eosinophil versus a monocyte, etc.
Okay, yeah. Are they still able to degranulate? Not sure. Obviously we do see the drop in the neutrophils, as I explained earlier. Whether that's because they've degranulated or not, I'm not sure. It's a good question, and it's something that we would have to look at probably in the future.
Okay, no problem. And just in terms of what's actually being used in terms of staining panels, they're all listed in our BloodReady white paper, which we're more than happy to share after the webinar. Question from Lu Zhang. What is the permeability of hydrogels for oxygen and nutrients? Will cells be under starvation or hypoxia conditions when they are encapsulated by the hydrogels? Would you like to take that one, Sarah?
Can do, yes. So because when the cells are encapsulated, the gel is stabilizing the cell membrane at those lower temperatures. And when they're at that lower temperatures, the rate of metabolic activity is so low, they don't actually undergo those processes. So they're not suffering from hypoxia or starvation because they're in this state of sort of hibernation. I'm not sure if you want to elaborate on that at all, Steve.
No, that's the simple premise behind it. You reduce temperature, you reduce metabolic activity, but usually cells die in that state. So the technology enables them to stay alive through protecting their lipid membrane, which is exactly what you said, Sarah. So I'll get back to asking questions. Another question from Ruth. Do you have plans to release blood collection tubes so that clinical sites do not have to do any mixing of the beads and blood sample? That's probably one for me to take. We don't currently have the capabilities to manufacture within vacuitainers for immediate clinical collection. It does currently involve a transfer of the blood to a different tube. But we do plan to look for partners to co-develop a product whereby the whole system can be manufactured within a vacuitainer for immediate collection. Okay, let me just scroll through. Okay, there's another couple of questions. Can PBMCs be frozen after storage of the whole blood? So store the whole blood and isolate the PBMCs and freeze them.
Another good question. Obviously, you saw within the data you're able to isolate the PBMCs after storage in BloodReady. We have looked at then refreezing PBMCs, as many people would do in their process. And you are able to freeze them for them and retrieve good recovery. We have also looked at this and compared freezing the PBMCs with our other technology CytoStore that I mentioned earlier. And for anyone that's interested in keeping those PBMCs at sort of hypothermic temperatures, CytoStore works very well in that process too. So depending on what your process looks like, you can freeze the PBMCs or you could use CytoStore to, for example, ship at room temperature.
Fantastic. Thank you, Sarah. I think we've just got time for another question. Would this work with bone marrow?
But potentially we have looked at we have successfully preserved stem cells, so hematopoietic stem cells, mesenchymal stem cells in CytoStore, obviously because it's just this specific cell type, where you're able to store them for up to 5 days at room temperature. Obviously the bone marrow compartment contains a lot of stem cells and also myeloid progenitors. So theoretically, yes, though we've not specifically done that, I would anticipate good storage and recovery.
Wonderful. And are there any further questions from the audience? Okay, no problem at all. Well, thank you so much for joining this webinar this afternoon. I think we've just got one last one in from Emily. Have you checked if plasma or serum generated after BloodReady stabilization shows any differences in proteomics or metabolomics analysis?
At this stage, we haven't managed to look at that. We've been sort of more concerned with the immunophenotyping profile. Obviously, that's an important outlook to look at as well. But we don't have any data at this point. Hopefully in the future.
Wonderful. Thank you, Sarah. And all the questions are flying in now. A question from Bill. Can you isolate plasma after storage?
Very similar to the previous question. We have only really looked at the PBMC fraction, not so much the plasma. So it's something we'd have to look at and see.
Thank you. Just to add to that one, we would assume that the plasma would be completely integral, but after addition of the dissolution buffer, you would anticipate a dilution of the plasma. But because everything is maintained in the blood, we would anticipate there to be the plasma compartment still able to be processed. And just a question over storage temperature from Aria. In the protocol for leukapheresis, 25 degrees is recommended for leukapheresis. Is it recommended to maintain this constant temperature? And what differences are there between leukapheresis and BloodReady?
Good question. So we, as mentioned before, we have Leukastore for the preservation of leukapheresis material. The difference in temperature recommendation is obviously because in whole blood you've got the complete fraction of everything, whereas in your apheresis material you've enriched for leukocytes and removed certain other components. And we know that T cells in particular are very happy at 25 degrees, also 4 degrees, but if you can store at room temperature, it's slightly easier than refrigerated. So for the leukapheresis material, we recommend 20 to 25 degrees just for the best possible outcome. Whereas the whole blood, 2 to 8 degrees, we know that monocytes and B cells prefer that lower temperature.
Wonderful, thank you, Sarah. And I think that's all the questions now. So just from my side, just wanted to say that if you were interested in trying BloodReady or any other of our products, we are able to offer a 25% discount on any of the products. And even more excitingly, our new plushies, RT plushies, will be dispatched with any order before 31st of March. I believe that information will be sent to you after the webinar, but if not, please get in contact with Sarah or sales@atelerix.co.uk, and we will be more than happy to provide you with any further information you require, or we're also happy to catch up individually if you have any further technical or application-based questions. With that, a very special thanks to Sarah for that presentation, and I wish everybody on the webinar a fantastic afternoon. Thank you so much.
Thanks, everyone.